Hi, I am Alice Peters and welcome to this latest video. In this playlist, which is all about bio-inspired materials, I'm going to talk about how we can use molecularly imprinted nanoparticles, also short nanoMIPs, for the detection of contaminants. And the first thing we need to talk about is why we need this bio-inspired design. So why do we need mimetics for antibodies? Well, the first thing to note is when we talk about antibody mimetics, we talk about structures that mimic the affinity of the antibodies, so having a high specificity and selectivity for your target molecule, but the structure will be completely different. And there are multiple options out there, so people have commercialized things like aptamers, affibodies, which is on well-powered molecular machines, but in this particular video I'm going to talk about how we can use molecularly imprinted polymers or MIPs for this, and specifically nanoMIPs. And there's a clear need for this because, for instance, these antibodies, they have limited stability in terms of pH and temperature. A lot of antibodies are unfortunately still produced using animals. So even though you might not see there's a lot of hidden animal use in animal-derived antibodies. And if you look into publications, you can see that it equates to 1 million animals per year used in the EU for these diagnostics. And then there's also like a cost issue, and it can also be significant batch-to-batch variation when you work with antibodies. So all of this can be avoided if you work with synthetic alternatives, and in this case I'll specifically talk about how you avoid this by using MIPs. In this very brief animation you can actually see what this molecular imprinting process looks like. So what happens? Essentially you have something that you want to detect, which is your target molecule or your template, and then you assemble monomers around it. So these monomers, they form these non-covalent interactions, and that's similar to your enzyme and your antibody, and that should be able to form like a pre-polymerization complex. You polymerize around it, so you form like a rigid 3D structure, in this case the polymer matrix that will keep binding sites in place, and then you take out your template, which means that you end up with a porous material which contains cavities that are similar to the size and the shape of your original target molecule, but also the chemical functionality. Now, if you want to predict the affinity of the mixture that you're working with, so predict the affinity of your polymer for your target, people generally tend to look at this pre-polymerization complex. So you can use characterization techniques for this, but if you would do some molecular modeling or some computational modeling, you can see that the binding enthalpy of that pre-polymerization complex can be indicative for the later affinity, and that's what people generally tend to use. Also, it's very important that we work with references, so the non-imprinted polymer, which is essentially the same as the process just shown, but without the addition of the template molecule in the beginning. These non-imprinted polymers or NIPs are often used as references, but you could also imprint, and that's becoming more and more common nowadays, with a molecule which is similar in size and shape. There's actually a long-standing history of MIPs. So the first paper was reported in the 1930s, where they showed that if you use different solvents, that the material that you use would behave differently. But the real thing when it starts to recognize molecules, so that there was a molecular memory, was shown with diene molecules by Dickey in 1949. Now, unfortunately, because all of this was based on covalent interactions, there wasn't really widespread use for this technology. But in 1993, when there was a Nature paper on using non-covalent interactions, this is when MIP technology really started to take off. And examples of molecular memory of compounds you can see here, for instance, which relate to the shape, but also if you think about the imprinting process, this happens. You have also biological imprinting, so imagine something hatching from its egg and then recognizing what was originally there. So when you talk about biological memory, this talks about the parents that they see, or in this case, whatever it sees first when it hatches. This is also similar to these molecules. So if you would have like a polymer, whatever it was in contact with when you prepared it, or when you synthesize it, it will recognize that later on. Let's briefly compare. So we have natural proteins and then we have synthetic mimics. So we know when we work with natural proteins, generally we have 23 amino acids that we can work with. Doesn't seem like a lot, but it pretty much does the job. So very defined sequence, very defined specific structure, and then you have weak bonds that really keep that 3D structure together. Now, what you do have to know here, we're talking about millions and millions and millions of years of evolution. So whatever has been around in terms of enzymes and antibodies, that's been optimized by so many years. If we look at the other end of the spectrum, so we look at the synthetic alternative, essentially you would have an unlimited library that you can work with. Even though you will see that a lot of the monomers that we're working with, they still pretty much resemble amino acids. So there's a reason why they are used. The sequence is much less defined, so the structure that we're working with is much less defined in general. And we have the cross-linking or the stabilized bonds rather than globular weak bonds. The advantage also is that we can really fine-tune the architecture. So we can use particles, we can use layers, there's lots of different variations that we can play with. We can also integrate things like fluorescent probes or, for instance, redox probes. So there's a lot of versatility in there. Obviously, we don't have millions and millions of years of data in optimizing our design. So we often need to use something like computational tools in order to do this. And we can use experimental design, but a lot obviously takes a lot. But imagine that, yes, if we would have like a tool that can really predict the best composition, we could very quickly optimize a MIP for a very specific target. And then because we have many more than just the 23 amino acids at hand, we can really work with very bespoke systems. And in principle, we should be able to at least rival the affinity of natural receptors. The typical process of making MIPs used to consist of just simple free radical polymerization where microparticles were produced. And often this could also be done as bulk polymerization, which essentially means that you don't necessarily add a solvent, but you dissolve it in, for instance, the monomers and the crystalline monomers, which can be liquids as well. So a very simple process, but obviously this leads to very heterogeneous particles. So that also means, besides that you have a distribution in the size, it also means that you get an affinity distribution, and those particles definitely cannot rival the affinity of antibodies. However, you can find these microparticles commercially. So for instance, if you look at purification and extraction, you're not so much worried about the selectivity and specificity, but it's more a binding capacity thing. And for instance, in Sigma Aldrich, you can find things around MIP, where you can look at extraction and purification of, for instance, certain pesticides or certain hormones. So this is a very important application of MIPs, but it's a very different way of looking at it, because when you're working with sensors, you want really very defined structures, and you need really, really high specificity and selectivity, which you don't necessarily need with chromatography. And therefore, there's been a move towards these nanoparticle systems. And if you briefly want to see what this general polymerization protocol looks like using things like free radical polarization, have a brief look at this, well, let's say 10-second video where you see how the polymer is actually formed. And you see it's a very simple process, and it's also a very scalable process, so you can do this on very big scale. And therefore, there's been an ongoing search into using other systems where we can make much more reproducible particles, but also those who are much more homogeneous and therefore only have a very specific affinity. And what you will see, if you have a way of selecting the ones with the highest affinity, then you can start to compete with, for instance, antibodies or enzymes. Now, people traditionally approach this, and this has been ongoing for a bit, using a solid phase synthesis approach. So here you flip it the other way around. So rather than extracting your templates, which can be very cumbersome, very time consuming, you can also extract out the polymers. So what people do is you use a solid phase, which can be as simple as a glass bead. And what you do is you attach whatever you want to detect to your template molecule, or if you're working with a protein, you could often use an epitope thereof. So you covalently attach this to a glass bead, and then you actually start to polymerize around that. And then afterwards you extract the polymers using usually temperature elution steps, and then you only obtain the ones with a high affinity. So people use thermoresponsive monomers, so something like, for instance, NiPAN, which changes its shape upon changes in temperature, and therefore you can collect them based on temperature elutions. So by flipping it the other way around, just by collecting polymers and using the solid phase as an affinity medium, you're able to really only target out the polymer particles that have a very high affinity, and you can automatically remove things that either are monomers that didn't react or only have a very low affinity because they come off at lower temperatures. So this process has been around for a while now. And what you can do is you can start to get very homogeneous distribution of particles, so things that are in the order, let's say 50 to 150 nanometers. That's what you typically see in literature. And the advantage of these is these materials are water soluble, so they're much easier to use, and you can, for instance, autoclave them, so you can really store them for longer periods. But it's very important because you're working with much smaller particles as well. It's shown that they are biocompatible. And that brings advantages in terms of if you want to look at MIPs for instance, therapeutics or other options that go beyond sensing. There are obviously a couple of complications with this approach. It's definitely much harder because you will need to covalently attach whatever you want to attach to the glass beads, which involves an additional step. Because we use the solid phase as in like an affinity medium, you will see traditionally the yield is much lower. So we're more talking about 5 to 10% or less. So that also brings some queries around the scalability of it. And in generally, it's just a more labor intensive process. And here you can briefly see how you can use something like Cylonization in order to quickly attach either your target molecule, which can bear, for instance, some carboxylic end groups, or some amine end groups to your glass beads, or if you're working with an epitope of a specific protein, for instance, which you can easily order with an additional SH group, you can easily attach that using other chemistry to these glass beads. So there's various options on how you can actually functionalize it. And these glass beads you can recycle later on as well, which means that you can keep on using them, and it really reduces the cost, specifically if you're working with proteins. So that's a big advantage of this. And what you would want to do first, there's a couple of assays which you can use in order to check whether you actually have peptides on the glass beads. So if you wanted to confirm that you've actually functionalized your peptide, you can see, for instance, in this assay, there's a very clear color change. So we know now that whatever we wanted to have on there is actually present, and then we can start our polymerization process. Now, typically we do this in our lab. We have like our own kind of synthesis setup where this is all done manually, which is very feasible. But obviously, if we want to look at towards commercialization, it would be better to have like an automated reactor system. And obviously, I did mention before that the yield is quite low. So if you could streamline the procedure, that might help in order to get a higher yield from this. So in essence, you can find some examples of some automated reactors in literature, but there's definitely a high demand in order to automate the system even further. And it is just a few things that you need to take into account. Obviously, you can use something like a UV jacket if you wanted to do UV polymerization. But this is a specific example where they were doing, and this is the more traditional solid phase approach, where we have water-based systems where we use a combination of APS and TMIT in order to kickstart your reaction. What they did there is things that are really crucial to understand is that we need to make sure that your vessel that you're working with is very well mixed. So that mixing is really necessary in order to make sure that all the compounds are in contact with each other, and that also leads to a more homogeneous system. And then we also need to have like some kind of temperature control, because we know we can only get the very high affinity nano-MIPs via this temperature-based elution. So you need to make sure that there's temperature control embedded in it, where we can do a wash at lower temperature. So we really need to cool it then, so we go actually below room temperature. And then we need to kind of heat it up to about 60 degrees, where we can take off the high affinity particles. So polymerization tends to be an exothermic process, so that's definitely one of the limitations of scale-up. And even though this is a relatively fast process, so let's say anywhere between 2 to 4 hours where you can have a batch, the dry yield, you're still talking about milligram scale, so you're not even going towards gram scale. So that's with this type of reactor which was presented, you can see this is still relatively low scale. But do imagine if we're working with sensors, you need almost like a minute quantity. So if you would have a milligram, for instance, of nano-MIP, this would get you a very long end with almost like an infinite number of measurements that you can actually do. So this is very important to, for instance, for the scale up, but there's still very limited options outside of the lab that we can work with. But imagine, let's say if you wanted to do high throughput screening, there's definitely a demand for systems that can more precisely control all of this, and particularly looking at controlled polymerization mechanisms, like for instance rafts, in order to get a more controllable nano-MIP structure. It can be pretty time consuming developing and optimize a MIP for a specific target. So the first question that you might want to ask, what is the best way of selecting certain monomers? So if you wouldn't have a lot of computational tools available, if you're working with a protein, you can go for a standard recipe to start off with. And here you can see a range of monomers that are actually used that cover different interactions. So you would have a combination of ionic, hydrogen bonding, hydrophobic interactions, and together that mostly covers everything that you roughly need. And obviously then you could play around with the ratio to see where you get at. So you could start off with that. But there's other tools that you can use. So I mentioned computational modeling before, and in the next slide I'll show you in a little bit more detail how we do this, for instance, in Newcastle. But there's also lots of analytical techniques that might help. So what you then do, rather than actually looking at the final polymer, so as I mentioned previously, you probably look at the pre-polymerization complex. So you look at what happens when you actually have your target in the presence of the monomers, and if you can detect anything there. So techniques that you can potentially use are ITC, where you can look at the binding enthalpy again and changes in temperature that you can correlate to whatever is happening in your polymer. There's a couple of other ways you can do. You can do some titration, so you can do that, for instance, using fluorimetric techniques, so detection of fluorescence. Similar NMR is also used, because often if there's hydrogen bonding, you can see shift in the protons. So you can also use that, for instance. But then besides those kind of methods, where I said like you can look at what different monomers you can use and can predict things, what's also very helpful there is that you can get an idea for the ratio. Because it's not just what monomer are you using. Well, ideally you would want to use it 1-to-1, but you very rarely get, there are some very bespoke monomers where you can do that, but usually you work with a little bit of excess of monomer. So let's say if you're working with a couple of different batches, there are some methods around where you use, for instance, like a 96 well plate in order to go for high throughput screening. So there's a couple of papers where you can look at, for instance, if your target would have like a free amine group, there is some very quick screening where you can make use of that free amine group in order to determine what's happening. And there's also papers where you find that there's direct replacement of MIPs with an antibody, so where you use an ELISA type of format. And you can do that because targets that they're working with their function life of HRP, so you directly have like an ELISA type of system. And then if you have multiple batches of polymers, you can very quickly test using some optical techniques, whatever works the best. So here's like an example of the computational modeling and some of the work that we've published with my collaborator in chemistry, which is Dr. James Dawson. So what you see here is where we look at one particular monomer, so in this case aniline, which has the advantage that it's electroactive. And then we looked at basically the binding at one specific binding site, so we kept that the same with a range of molecules. And we were interested in detecting levodopa, and then we could see some of the molecules that were quite different. The binding energy was very different, so bearing in mind a lower binding energy, so a more negative value is better here. But you could see for another compound, dopamine, which is actually quite similar compared to L-dopa, you could see that the binding energy was almost the same. So what the computational modeling told us here is that we really wouldn't be able to discriminate between those two molecules, and that actually was in agreement with the experimental data. In order to do this, the first thing you've got to do is you need to identify the binding sites. So you would need to you need to look at the monomer that you've got and think like, well, we kind of know that, for instance, if you've got a carboxylic acid or an amine, that will facilitate binding. And then we also need to look at the binding places on the molecule. And if you're working with a small molecule, that's relatively straightforward. It does become a lot more complicated when you're working with a protein. And particularly because we don't know where all the binding sites are. If you work with an epitope, then you would need to look at the binding to, for instance, one amino acid residue. So much more complex. But in essence, what you do is step one is you identify the binding sites. And then in step two, you need to kind of optimize the binding between your target and that binding site that you've identified. So that means the bond lengths and the angles and things like that. And then the third step is you need to calculate this binding enthalpies, and you can compare them against each other. And while this seems relatively straightforward, I would say, it does become a lot more complicated with mixtures. And it definitely becomes a lot more complicated when we're looking at proteins where we've got so many binding sites. and you don't know exactly where your monomer can potentially interact with multiple different binding sites if you've got a protein, and you don't know exactly where it's going to bind. So for small molecules, this is much better understood and therefore somewhat easier to model. Another major advantage of working with polymers compared to, for instance, antibodies is that these polymers are much more versatile and much easier to adapt. I mean, you can easily buy an antibody with like a fluorescent moiety. We do kind of know that. You can easily buy them with a label. But if we work with the polymers, you have much more scope of kind of really changing the structure and embedding in extra functionalities that you might like. Well, the key things probably people use are still like an optical kind of probe or like a redox probe, and they're usually done at like 10% of the mixture. So these labels are not necessarily there for the binding, so they actually don't really help with the binding, but it's more for visualization. And that's quite straightforward. And some of these, particularly the optical labels, there's very standard techniques for how you can integrate those into your polymerization mixture. So fairly straightforward. There's also thought of potentially altering structure afterwards, which might give you more control over the structure. But then it opens up a whole other scope of materials that you can make. So for instance, people look at magnetic nanoparticles. And what's also been growing in popularity is the so-called double imprinting approach, where we're not limiting ourselves to one target. So you can find very interesting papers where they look at imprinting, for instance, with like an epitope of a protein, so a protein which is present like kind of cell receptor, but then also imprinting with a drug molecule where you can start to look at drug delivery. But the drug delivery is then more like of a passive process, and you don't have both of the targets actually in the solid phase. You only have your protein on the solid phase. So that does give some limitations. What you can also do, and what's quite interesting, is you can do like a double polymerization step. So you can have whatever you want to detect first, and then you kind of polymerize around it. And then when that's still locked into place, so before you actually start eluting off the high affinity materials, you can do a second polymerization step. So you can add in a dual functionality. And that can be things people have done with magnetic nanoparticles, which can be quite interesting. You can add like an additional conductive layer perhaps, or you can add things like enzymes to it, where you can find things where people not only look at binding to cells, but then also incorporating, for instance, an enzyme that removes sugar groups, so can also be very good for killing off the cells and is therefore like a therapeutic agent. So there's lots of opportunities there with nanoMIPs that you potentially don't have with antibodies, and I think we're only just starting to explore what we can do there. So let's go over a brief summary what we can actually achieve with these molecular imprinted nanoparticles, or the nanoMIPs. Well, I would say they can definitely rival the affinity of antibodies, but they have advantages such as they're very cost effective, they're very versatile, they're very robust in terms of withstanding pH and temperature, and of course this is completely animal-free technology, so it does have some ethical aspects around there as well. They are very widely implemented in research, but in commercial senses there's no applications out there yet. I think that will happen very soon, but barriers that will need to be overcome, so we need to find really like one killer application where we can use this for, and where they're actually either better than antibodies or replacing them in areas where we don't have antibody-based sensors yet. Now, other things that really need to be looked at is how we can produce them on a big scale, because definitely the yield of the solid phase approach is quite low, and how we can do a kind of like a one-step integration of these materials into sensing. For instance, if we would do some printing procedures, you can directly integrate the sensors with the polymer materials, because for now we have to do multiple steps in order to covalently attach them, which is obviously not very convenient. Another big advantage is what we'll see with these polymers is that they can have these dual functionalities. So we can really see that we can really start to exploit that, and are very easily modified with, for instance, like redox and optical probes in order to visualize them. And so there's a lot of scope for these molecular imprinted nanoparticles, and let's see what the future holds. Perhaps we will be able to replace all antibodies with nanoMIPs in the future. Thanks for watching, and if you do really want to know more about these bio-inspired materials, then do have a look at this playlist.